Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Mitinori Saitou from Kyoto University on this show. Please let me briefly introduce you to our audience. You received your MD from Kyoto University Faculty of Medicine in 1995 and received your PhD in 1999 from Kyoto University Graduate School of Medicine. You then moved to the Wellcome Trust Cancer Research Campaign Institute, where you worked as a postdoc in Azim Surani's laboratory. Then you were appointed team leader at the Riken Center for Developmental Biology in 2003 and appointed professor at the Kyoto University Graduate School of Medicine in 2009. You then became director of the JST Erato program in 2011. You were then appointed professor at the Kyoto University Institute for Advanced Study and director of the Institute for the Advanced Study of Human Biology in 2018. And in 2024, you were appointed program officer of the JST Forest program. A question I like to ask every guest to start off our little podcast is, how did you become interested in biology in the first place and then in pursuing a career in science?
I think as many of the boys are, I was kind of very much interested in insects and living creatures like fishes, and played a lot for catching them. So I think I have long been fascinated or interested in how our world is kind of organized or kind of created, and what the lives are, or something like that. So basically, I was kind of very much interested in how we come, our origins and our neighbors, and how the living things are kind of created during the long history of evolution or kind of life.
Mm-hmm. So it was biology from the start, basically.
Yeah, exactly. It's a very, not really, medicine based, more like a biology based.
Mm-hmm. Okay. So coming to your science that centers around the mechanisms and reconstitution of in vitro germ cell development in mice, non-human primates, including great apes and also humans. In the beginning of your research career, you focused on characterizing the function of two proteins. It was BLIMP1 and PRDM14 in the germline. So to ease into the topic, can you maybe talk about what you found about the function of those two proteins in the germline?
Yeah, so I took a kind of very, long time to think about what sort of themes I'm going to pursue during my scientific career. And as I said, I was very much kind of interested in the, life and living creatures, and how we, come from these primitive, living organisms through kind of evolutions. I thought, and also I was, as a kind of medical student, I was sort of very much interested in why, individuals or humans are kind of die. So I was kind of thinking these kind of things, and thought it might be very much worthwhile to study the biology of the germline or the germ cells, because they are the origin of kind of everything, and also, in a sense, immortal, to perpetuate the continuity of the life. So I decided to work on the mechanism of germ cell development. And when I started, it was late 99, it's the very end of the 20th centuries. And because I was having a medical background, I wanted to work on mammalian systems. And then I decided to, analyze the mechanism of mouse germ cell specification. And one thing that fascinated me was that there was really nothing at the molecular level known about the mechanism of mouse germ cell specification. And they are using in a sense, very primitive technology of a single cell cDNA differential screening. We have succeeded in identifying key genes important for mouse germ cell specification. That was, as you said, the first one was the BLIMP1. And the second gene was its close relative called the PRDM14. And they are the transcriptional regulators wearing so-called PR domain and also gene fingers. So it's a transcriptional regulators. It doesn't have, they do not really have a kind of histone methyltransferase activities, although they have this PR domain. But they have very kind of interesting functions. For example, BLIMP1 was found to be, so by a careful kind of transcriptional analysis, we found there occurs 3 key events upon specification of the germline in the mammals. So it's a segregation from the somatic lineage. So the one event is kind of reacquisition or re-upregulation of a potential pluripotency network. And the second is like a repression or suppression of the somatic mesodermal program. And the third is, triggering subsequent epigenetic reprogramming. So germ cell specification consists of these 3 key events. And if you knock out BLIMP1, everything is just disrupted or impaired. Nothing happens. And if you knock out PRDM14, then the acquisition of pluripotency and also ensuing epigenetic reprogramming is kind of impaired. So BLIMP1 seems to be important in the case of mice for all the events for germ cell specification, while PRDM14 at least seems to be important for the 2 critical events. So that's kind of our findings through the transcriptome analysis in the knockout animals.
So when you want to get human-induced pluripotent stem cells, there are the Yamanaka factors, right? And what you also did here is, like, if you overexpress BLIMP1, PRDM14, and a third factor, then you can kind of do the same thing, but for the germlines, right? Can you talk about this in a bit more detail?
Yes. So this is somewhat different from like what Yamanaka-san has done, because we are more kind of developmental biologists. So in a sense, we think the context is kind of important. So the cell fate is kind of established upon, gradual changes or progressive, changes of cellular state, which we call kind of competence. So what we have done is that we have overexpressed these 3 key factors: BLIMP1, PRDM14, and at that time TFF-AP2C or AP2 gamma. If you overexpress these 3 factors in embryonic stem cells, then very aberrant cell types are kind of induced. They overexpress some genes, some germline-related genes, very kind of highly, but not really upregulated other ones. So we thought this kind of cellular context is important. So we know that the germ cells are induced in their precursors called epiblast-like cells, or the kind of epiblast. So now mouse ES cells are kind of currently considered to have a naive state of pluripotency, but the post-implantation epiblast or epiblast-like cells are known to have a formative state of pluripotency. So we overexpress these 3 factors in this formative state, then these cells directly differentiate into primordial germ cell-like cells that contributed to spermatogenesis upon transplantation into testis.
So And now it gets a little bit complicated for me because these are really hard things to pronounce, but let's get into it. Next, you showed that human-induced pluripotent stem cells differentiate into incipient mesoderm-like cells, which robustly generate human primordial germ-like cells that can then be purified using some surface markers. Can you talk about this process, what you found in this study, and how this moved you down the path to your next advancements?
Oh, okay. So it's more than 10 years, I think nearly 15 years, or more than 15, like 10 years before, we showed in the mouse that mouse embryonic stem cells having naive state of pluripotency can be induced into epiblast-like cells, which are having formative state of pluripotency. And these epiblast-like cells, or EpiLCs, can be induced into primordial germ cell-like cells in response to BMP4. So we thought maybe we can, based on this kind of finding in the mouse, we wondered whether we can induce human iPS cells into PGC-like cells. We know that the human iPS cells are kind of more in the state of so-called, prime state of pluripotency, which is somewhat also different from, maybe more advanced than their formative state. So we are not really sure whether we can really induce human germ cell-like state from the conventional kind of iPS cell culture. But so initially we directly tried a kind of direct induction of human iPS cells into the germ cell state just by stimulating with BMP4 key cytokines. And what we have found is that we somehow surprisingly see low-level induction of PGC-like cells upon direct stimulation with BMP4. So we thought maybe human iPS cells have a kind of capacity to form germ cells, unlike mouse epiblast stem cells. But maybe the condition was kind of not really perfect. So we wanted to, differentiate it a little bit toward, precursory state of having a kind of mesodermal state. It's a stimulation of by activin and Wnt. And then that state, although it is still very much similar to iPS cell state in terms of expression of pluripotency genes. But this stimulated IMLC, or incipient mesoderm-like cells, then upon stimulation by BMP4, are induced very efficiently into the PGC-like cells.
So what we on this podcast are obviously interested in is epigenetics, right? And,
sure.
And this is then leading into my next question. You looked at chromatin remodeling during germ cell specification from mouse embryonic stem cells. So which marks did you look at and which changes did you see during this process?
So, of course, we initially see DNA methylation. So as you know, during the epigenetic reprogramming in the mouse, nearly all the DNA methylation in the genome are kind of erased during a 4-day sort of period. That was really kind of amazing process. And also at the same time, histone H3 lysine 9 dimethylation is also somewhat erased. And instead, histone H3 lysine 27 trimethylation, H3K27 trimethylation are kind of upregulated. So DNA methylation and H3K9 dimethylation erased and H3K27 trimethylation upregulated. And also probably the level of H3K9 trimethylation is somewhat decreased. But in the case of the mice, I think the key change is for these 3 or 4 modifications.
Mm-hmm. Does that then mean because DNA methylation is like a repressive mark, right? And
yes.
Does that mean that all genes can or will be expressed? Or is it just like erasing the memory and then reestablishing?
So more recent study have shown that all DNA methylation are somehow nearly all, except for some those around the evolutionarily young transposable elements are kind of all just gone, just really erased. But there's no event such as hypertranscription happens.
Mm-hmm.
This seems to be, and also what we have found is that the mouse PGCs are kind of also upregulating H3K4 monomethylation. So it's really, how they say, accessible, a prime state of kind of transcriptionally very permissive state with no DNA methylation. But such sort of hypertranscription doesn't kind of occur.
Mm-hmm.
And one of the reasons, at least in the mouse, is that the upregulation of H3K27 methylation. That really marks the promoters of many genes and kind of make it a kind of very repressed state.
Mm-hmm.
And the other finding that we have recently done is that somehow these reprogrammed chromatin, although they are transcriptionally very permissive, are highly insulated. So there's the 3D structure of the chromatin is somewhat, how to say, insulated very strongly by kind of cohesions,
Mm-hmm.
so that the strange enhancer promoter interactions are kind of prevented. So
Okay.
it's a very interesting system. No DNA methylation and the H3K4 monomethylation upregulation. So it's really transcriptionally poised all over the genome. But the key elements are repressed by polycomb H3K27 methylation. And also in the 3D genome level, it's highly insulated so that the strange promoter enhancer interactions are kind of prevented. So that's state of reprogrammed germ.
And if you would do ATAC-seq, would you see like more peaks?
Yes, more peaks. It's a very open state.
Mm-hmm. Yeah.
More peaks in ATAC-seq and H3K4 monomethylation, highly upregulated state.
What you then did was to expand mouse primordial germ-like cells in vitro and investigated their epigenetic state. Can you talk about this experiment and what you found there?
So that's what I have already talked is the finding from this expansion.
Then you show that human primordial germ-like cells differentiate progressively into oogonia-like cells during a long-term in vitro culture experiment. And this takes approximately 4 months in xenogenic reconstituted ovaries with mouth embryonic ovarian somatic cells. Can you talk about this study and what the breakthrough of this study was?
Okay. Initially, so the epigenetic reprogramming in human germ cells occurs, initiates just after PGC specification. And after PGC specification, they migrate through emerging hindgut endoderm and then go through mesentery and then colonize embryonic gonads where they kind of very actively proliferate. So epigenetic reprogramming happens during these 3 or 4 weeks period. In humans, we could induce human PGC-like cells, but these cells didn't really show epigenetic reprogramming. So we just wanted this reprogramming to occur in this human PGC-like cells. And the first strategy we have took, which is also 5 or 6 years before, is that to reaggregate human PGC-like cells with mouse embryonic ovarian somatic cells to see what may happen. So we didn't really expect anything may happen because we thought it's like evolutionary very distant human PGC-like cells and mouse ovarian somatic cells. But we just kept culturing them. And then we somehow found after like 3 or 4 months, the PGC-like cell derived cells still kind of survive in this embryonic ovarian mouse ovary structures. And then somehow started to express key genes that are activated upon DNA demethylation. I mean, epigenetic reprogramming. So then that includes genes called DDEX4 or Basa hormone or DAZO or some other so-called germline genes. And then we found not just these cells express these germline genes, but they also erase genome-wide DNA methylation compared to just induced human PGC-like cells. And these epigenetic states are somewhat very similar to what was reported in the human orgonia or mitotic prospermatogonia-like cells that kind of people analyzed from like aborted human embryos. So this is the first part. So we succeeded in inducing epigenetic reprogramming and then orgonia or mitotic prospermatogonia-like cells in xenogenic reconstituted ovaries. But this is somewhat very inefficient and also not non-natural process. And everything happens in this black box. So once you generated reconstituted ovaries, then you just put it in culture, and then you cannot do anything other than like a medium change. So and also the efficiency is somewhat low. When you start with like 5,000 PGC-like cells, after 3 or 4 months, it end up with like 500 orgonia-like cells. So the number is like decreased in 1/10. So and also the regulation of this system is somewhat very difficult. So we wanted to induce PGC-like cell differentiation under more defined condition, like without using gonadal somatic cells. Mm-hmm. This is what we have very kind of recently sort of published. So we have performed a kind of screening of various cytokines or chemicals to see what factors may upregulate some of the genes activated upon epigenetic reprogramming. So we performed this kind of screening and found that somehow, somewhat unexpectedly, BMP4 or BMP2 or maybe 7, if you continue to provide them for PGC-like cell culture, then after like 2 or 3 weeks, some of the very early activated epigenetic reprogramming responsive genes are kind of upregulated. So we thought maybe BMP is a kind of key factors that differentiate PGC-like cells into like orgonia or maybe mitotic prospermatogonia-like cells. So we, fine-tune various kind of culture conditions and eventually found that the, as reported very recently, if we culture human PGC-like cells on some sort of feeders with BMP, then they continue to, expand, propagate, and at the same time differentiate and also induce epigenetic reprogramming to become orgonia or mitotic prospermatogonia-like cells. And it happens in like 2 to 3 months. So it's somewhat the time course closer to what you see in the embryos.
So the study you were just mentioning, is this the Nature paper from May this year?
Oh, yes, exactly.
Yeah. Okay. I had some questions before that, so maybe we can go back 1 or 2 years. What you also did is you went back to human-induced pluripotent stem cells and investigated their epigenetic state or signature when comparing different human-induced pluripotent stem cell lines. So exactly, what does it mean, like that there are different lines? Is it like their origin is different, or what does this exactly mean?
Yeah. Okay, origins are different, and also different sexes, like male or females.
Okay. And what did you then find by comparing those?
So one of the things that we have found is that because we have somewhat wanted to induce like oocyte-like cells from PGC-like cells, and that means you have to start with XX female iPS cells. But it has somehow been known that the female human iPS cells or ES cells having a very abnormal X chromosome epigenetic state. So it's usually one of the X chromosome is kind of inactivated and one is kind of activated. But in pluripotent stem cell culture, one inactive X chromosome get, X eroded, or become very strange epigenetic structures, states. And also this kind of state also really depends on the culture conditions and also different lines. And so one of the things we really have found is that the iPS cells, or maybe even, this is already also reported by many others, but the X chromosome state really abnormal in pluripotent iPS cells. And we think it was really crucial to make a very good control of kind of this state if we really want to make good-looking oocyte or maybe orgonia or oocyte. So that's why we really wanted to study this epigenetic state. But other than the X chromosome, there are also variety of chromosomal regions that epigenetically abnormal or at least different among the different lines. And they could be, because, iPS cell reprogramming, if you can induce this pluripotent stem cell state, then various epigenetic abnormalities in the chromosomal regions, not critical for this maintaining pluripotency, are usually okay. So iPS cells are having lots of such line-dependent, strange epigenetic phenotypes.
Mm-hmm.
And that's, I think, a potential, problem for making high quality, maybe germ cells.
Mm-hmm. So the overall goal of your work, is this getting then human embryos out of stem cells?
Overall goal? So, one of the reasons why I was pretty much interested in the germ cell biology is that the one thing is the mechanism of epigenetic reprogramming, which is, I think, still not really fully understood.
Mm-hmm.
Our study points to the role of, for example, with regard to a DNA dematuration, the main mechanism is like a passive DNA dematuration. Because in the primordial germ cell-like cells, key active, DNA methyltransferases like DNMT3A and 3B are kind of repressed. And also key factors for DNA maintenance methyltransferase, such as UHRF1, is also kind of repressed. So not really. There's only little active DNA methyltransferase machineries remaining in germ cells. Then upon kind of propagation, they progressively lose genome-wide DNA methylation. So mainly passive. But some genes requires active DNA demethylation, such as like DDEX or maybe DAZO. These are germline genes. And also maybe genes critical for meiosis may also require such sort of active mechanisms. But in humans, this is not really known. So because we have now in vitro system that really recapitulate human epigenetic reprogramming in the early germline development, we can really precisely analyze the mechanisms in a kind of detailed manner. That's one of our kind of interest. And then after epigenetic reprogramming, in case of like oocyte formation, the meiotic recombination happens to make a kind of oocyte. And this is also a kind of shuffling of genetic materials to create that kind of diversity for genetic information. And also, this mechanism is probably not well understood, at least in the humans. So because there's no nice systems, although currently there are many kind of single cell technologies to analyze what may happen at the genome level, but the mechanistic level, it is still, I think, not really easy to tackle. So then if we have a nice system for making like meiosis to happen, then we can also analyze this critical event in germ cell development. So one of my interests is to really establish in vitro germ cell developmental system, maybe in humans, and to really analyze the key mechanisms that underline successful human gamete development. And I think this would be probably useful for thinking about like etiology of like critical diseases, including infertility and other kind of genetic or maybe epigenetic abnormalities. So not just creating how to say germ cells, so making sort of human embryos. But we are very much interested in underlying basic mechanisms that support continuity of kind of our species.
So you just mentioned that some of the genes are not passively like erased, but they are somehow
DNA methylation.
Yeah, they need to be actively erased.
Yes.
Is there some protection mechanism, or how is this specificity then achieved?
Yeah, that's really a key point, and probably in humans, no one has yet analyzed, but probably like H3K9 trimethylation that is imposed at an earlier phase of development prior to germ cell specification, probably protects these genes as well as like evolutionarily young transposable element to get, how they say, easily demethylated in the early germ cell development.
So you just talked about your overall goals, but imagine you're due to submitting a grant proposal tomorrow. What would you have written into this? So you don't need to disclose any details, but just like the overall things that you want to achieve in the next 5 years.
The first sentence, maybe I still write to understand the mechanism of human germ cell development that will lead to treatment of some sort of diseases, including infertility or genetic or epigenetic disorders. So yeah, that's I will write.
Okay.
Not really directly, how to say, like to make like sperm or oocyte.
But rather getting behind the mechanisms of what defines the development of those cells.
Sorry?
But rather getting the mechanism behind these processes.
Yeah, exactly. Yeah. That's I think probably very important. Without really understanding this process and the properties of the cell, I think it is nearly probably impossible to create kind of human embryos.
Okay. I see.
Yeah.
So for the last 35 minutes, we have been on a journey through your scientific career. Did we miss something important, or would you like to add something?
Not really. I so far, how to say, enjoyed my scientific career very much. How to say, very fortunately or luckily, I can continue to pursue what I wanted to do when I started postdocs. And then I got very nice colleagues and also postdocs and students to continue or to create the area now called kind of in vitro gametogenesis. And this area is not just for creating kind of gametes for embryo production, but it's, as I sort of explained, really make a kind of foundation to analyze key mechanisms underlying germ cell development, which is really critical for like any, how to say, life mechanisms, I guess, because we all originate from like germ cells. So
obviously.
yeah.
So my last question, and this might be especially interesting to get the answer to this question from you, as you have a very special viewpoint to this. What is your personal definition of epigenetics?
Personal definition of epigenetics. Yeah, so it seems that originally this epigenetics comes from the term, this epigenesis, by like a Conrad Waddington. But epigenesis just means, in contrast to like a pre-formation, where everything is like formed initially, development happens by a conjective induction or something. So it's really a modern developmental biology concept. But maybe after identification of like key molecules, like modifying DNA or maybe histone proteins, then such sort of modifications that regulate how to say cellular phenotype are considered as kind of epigenetics. But personally, I haven't think about this very.
No, it's okay. It's okay if you don't have a clear answer right now.
Both definitions are just right and interesting. But many of the epigenetic things are in fact controlled by genetics.
Mm-hmm.
So in a sense, it's an epigenetics. I think is also someone, for example, say that for example, you can induce iPS cells from like somatic cells by just expressing transcription factors. So then people start to say, oh, it's just transcription factors that are important. So epigenetics is, in the end, not really critical.
Mm-hmm.
But I don't think so. So because, DNA methylation or histone modifications or other, how they say, so-called epigenetic phenomenon are really a kind of good system to really, how they say, make diverse cell types, or maybe to make a sort of state much more stable compared to without having such sort of modifications. So it's really epigenetics, so-called epigenetics. It's really a good invention or a cellular system that lock a certain state in a relatively stable manner, although perfect.
Mm-hmm.
So I think to understand this kind of stability, the mechanism behind this kind of stability and also sort of inheritability of the cellular state, maybe the epigenetics.
Okay.
And the germ cells, what makes germ cells kind of unique or special is that they are naturally really reprogramming or resetting nearly entire this kind of epigenetic memories to make a sort of new generations. And this actually, occurs twice during so-called germline cycles in primordial germ cells and also after fertilization in early embryos. So there's really two phases of this kind of epigenetic reprogramming to make, how to say, embryos really prevent inheriting some sort of epigenetic abnormalities. So I think the germline development is in a sense very kind of interesting to really understand how these so-called epigenetic marks are erased and added and also erased and added.
Mm. So thank you for your time and for being on the show.
Yes, thank you very much.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening and stay tuned.